Bom dia.
Good morning. It's a great pleasure to be here with you. My name is Fabrício Marchini. I'm a researcher from Fiocruz, Paraná, Institute Carlos Chaga, and I'm the manager of the Immunological Institute of Paraná, IBMP. I'm here to tell you about the development of thrombin and the biological uh, development. We have been developing diagnosis kits and other therapies through the years, and I'm here to tell you about the story of thrombin. Just to contextualize thrombin in biology, thrombin, just a minute, I'm trying to use the clicker here. So thrombin is one of the molecules of the blood circulation. Here you may see in details, but I won't speak about the different enzymes and the blood factors, cofactors. So thrombin is here. We have the activated thrombin, and we are developing a molecule based in a eukaryotic lineage that needs this eukarine. That was our choice to transform prothrombin into thrombin and generate this recombinant protein uh, based in eukaryotes. So it's a thermostatic agent, and it has as its objective to improve coagulation, preventing bleeding during surgeries, with the objective of improving clinical results, uh, saving hospital resources and time in hospital. The surgery time, we are able to reduce it as well. And it also aims at simplifying the use of hemostatic agents. There are several sealants in the market, and they are very different. We have synthetic hemostatic agents. Some of them are described here. They are absorbed. They are not active in this coagulation process with this solid matrix. So what we have in reality are different compositions. For instance, collagen with other sealants and other microbiological agents. And here we have the fibrin sealant and thrombin sealant as well. We also have different contexts in terms of protein origin. So here I detail different providers, different suppliers, and the origin of the different molecules. So I'm going to give you more details in the next slide. But we are talking about the development of a product that is similar to another thrombin product that is in the market. We started developing the thrombin sealant without the emergence of another thrombin in the market. But over time, this product became available some years ago, which is basically a recombining protein, also based on show cells, but they are mainly based on plasma, human plasma and ox plasma. That's what we are trying to prevent, the use of recombining technologies to have a better production. So this is the first sealant, the first biological sealant that is very similar to the molecule we are developing. This molecule is to replace the use of plasma, of thrombin derivated from plasma, from cows and from humans. And in order to control the productive process and prevent the use of proteins derived from this origin and with possible virus and other pathogens. So this is a growing market, not only a market of hemostatic agents, but the use of recombining molecules. We are replacing then this derived, this plasma-derived products to this recombining molecule. Speaking about our process, our development, it was made within IBMP, and we started with this knowledge and development chain from this learning curve with this expression system that is owned, where thrombin is based, is expressed in this system. This expression system is then transfected to this show cell, that is the producing cell, and we have the superspessor population advancement with this cloning methodology so that we can have this clonal population that is very expressive. So we have the densitometry to show the purification in the different populations with the expression quantified. After this process of selecting this super expressor clone, we have developed the upstream process of thrombin and generating these expression curves, getting over 10 to 7 cells with almost 20 days of expression, a little less than 7 days in reality. So this is the downstream process of thrombin, some details. After this fed batch at this system, we have the deep filtration, we have TFF and 3 cleansing steps. As I've mentioned in the beginning, we have to activate the prothrombin, and the way as we do that in this system is by using hecarin, and this is used to activate. So in this matrix, you're able to see the activated thrombin to the right. So after this process, we also increased its scale. We had 30-liter cultivations, but we are now changing the process in order to adequate its purifying process to make it compatible. So I have mentioned that before. This is the purifying step based on this cadence system with different columns. So we diminish the amount of volume of resin that we must use using several columns at the same time and obtaining a yield that is very interesting and with great purity. So outside of the upstream and downstream processes, we have fresh results in terms of functional analysis during the pandemic. So we've been working with some functional analysis of the enzymes obtained in the bioprocesses that I just mentioned. The first one is the functional analysis showing the activation of fibrinogen. So we have this capacity that is compared to this commercial commercialized molecule with this fibrogen matrix. We have a new picture that is the factor 13 showing the activation of the factor 13. So this is intrinsic to the activity. So we must show that. And we have the electronic densitometry showing the matrix generated by the recombining thrombin. Another data on functional analysis is obtained by Rotain. We show the coagulation time, and we are able to show that Well, we have specific data in this equipment, but we are able to notice that the combination of blood without recombining a thrombin, uh, ours and the competitors, they have similar times. We have activities related to the ability of irritating cutaneous tissue. We have this experiment showing that it doesn't diminish the skin viability, and it almost doesn't produce cytokines. So it doesn't have the ability to irritate the skin.
Esse é o último resultado que a gente obteve.
So this is the last result that we got, which is an analysis based on a rabbit. And here what we show is clotting time and the volume of clotting in a rabbit. So this is an experiment that's done with rabbit liver. And the results are here, as you can see. And clotting time and clotting volume are lower than if you didn't have a matrix. And here we have an additional result. It's not just thrombin, it's thrombin with another matrix, another gelatin solid matrix. And that shows that that's also important. And some of its uses are not spray-based. They use another type of solid matrix. And so it shows that this product can be used in this way as well. So basically, these were the last results that we obtained in this last phase of TRL3 and proof of concept, as well as part of the scaling process. So we conclude that the first results show that the product is not cytotoxic. It has activity proved in vitro with all of the examples that I showed you, such as the activation of fibrinogen and clotting time, and it's also effective in an animal model. So this is the conclusion of these phases of the process and not of the project. And our expectation now is to go to a non-clinical evaluation and do a clinical and enter the clinical phase in about 2 years. So that's a little bit of what I want to tell you. We're excited about this specific enzyme. This is the first molecule that's developed in-house, and we hope that to take the next steps soon. So it's an enormous pleasure to be able to share this with you. So that's it. I would like to stop here, and I'd like to open the floor for questions. Okay, so let me get the questions in case you have them. So if you have questions, you can raise your hand, or if you're watching this online, I will get the questions from somebody else. Thank you for your presentation. It was very interesting. And this is very important for development of diagnostic kits. But my question is related to validation and the validation of diagnostic kits. Can you tell me what were the parameters, the validation parameters that you used to ensure the quality of the diagnostic kit? Well, thrombin as a biological sealant, basically we did a comparison to commercial products, and knowing the cascade of clotting, we checked different molecules. So we showed fibrinogen being activated, we showed factor 13 being activated, which is what we hope in the clotting cascade. And we also showed clotting in blood in terms of clotting time and the size of the clot being formed. And we also showed the experiments in rabbits. We did this to show that using this product in vivo, you were able to stop bleeding. And so these are the experiments we've done up until now. And the sequence of these experiments and heading to validation in a clinical setting, basically what we have to do for thrombin is basically have 4 groups of surgeries where you're using thrombin and you're measuring the reduction of bleeding and the reduction of blood flow. So what we did for the Baxter molecule was basically we used 170 surgeries in 4 different groups, in 4 surgical groups. And we did this to show that that molecule was efficient in human beings. So that's a little bit about what we did and what we'll have to do to register this new molecule with the regulatory agency. So thank you. Another question? Okay, seems like we have another question. Hi, thank you for your presentation. So my question is kind of technical. So you use a purification tag in the process, right? It's probably a cysteine tag or something like that. So my question is, did that influence your validation? Did you need to remove this tag, or did it not change when you did something with a different supplier? No, there were no changes. So we were planning to do this validation with thrombin up until this moment in time. And since now we have a production process, and since we have an approved methodology, then we can remove the tag. That's a choice of ours. So we would like to have a clone with more expression. Our clone is expressing less than 1 gram per liter, around 800 milligrams per liter. We'd like to have a little bit more than that. We'd like to have a higher number, even though it's not an antibody, even though it's just a protein and naturally has lower expression. But we will do change. We will effect change so as to remove the tag and have more expressive organisms. So thank you very much. Have a great day.